Hello everyone and welcome to the biology lecture series, and today we'll be discussing the topic reproduction. So in the previous lecture I've given you a brief idea about the reproduction. Then we studied about the type of reproduction. So there are two types of reproduction. First one is the asexual reproduction and another one is the sexual reproduction. Then we studied about the type of reproduction, and then later on we studied about the sexual reproduction. So within the sexual reproduction, we first studied about the sexual reproduction in the plants, where we studied first about the structure of a typical flower. Okay, so then we were studying about the different whorls of the flower, that there are 4 whorls in the flower. Okay, and then we were studying about the pollination. So in the last lecture, I've told you how can we define the pollination, what is the pollination. So it is the transfer of the pollen grains from the anther, which is the part of the stamen, okay, to the stigma of the flower. So stigma is what? Stigma is the part of the pistil, which represents the gynoecium or the female reproductive part of the flower. Then I've already told you that pollination can be of the 2 types. First one is the self-pollination, another one is the cross-pollination. Okay, so this transfer of the pollen grains from the anther to the stigma can occur within the same flower or from one flower to the another flower, which is present on the same plant or on the another plant. Okay, this is clear. So this transfer of the pollen grains can take place from, or it can take place within a flower only. Okay, if we talk about bisexual flower, so it contains both the male as well as the female reproductive organ. So here the transfer of pollen grains can take place to the stigma of the same flower, within a flower, or you can write the same flower. Then what can happen? This is the first case. In second case, what happens? The pollen grains can be transferred to the stigma of the another flower. Okay, so this another flower, again, this flower can be on the same plant or on another plant. Okay, so these are the cases that can take place during the pollination. Either the pollination can take place in the same flower or another flower, and that another flower can be present on the same plant as well as on the another plant. Now first let's talk about the self-pollination. Now it is the process of transfer of the pollen grains from the anther to the stigma of either the same or genetically similar flower. This is known as self-pollination. Okay, so here the pollens are being transferred to the stigma, obviously, of the flower that has the same or that is genetically similar. So genetically similar flower will be present on, you can say, same plant. Okay, it will present on the same plant. Okay, or self-pollination can also take place in the same flower also. It can take place in the same flower in the case of bisexual flower. So these bisexual flowers, they contain both the male as well as the female reproductive part. Clear? Now self-pollination can be of 2 types. First is the autogamy and next is the netogamy. Okay, netonogamy. Autogamy. So auto means self. So here what is happening? So it is a type of self-pollination in which the pollen grains are transferred from the anther to the stigma of the same flower. Okay, this is known as the autogamy. So this takes place in flowers of wheat, rice, okay, as well as the pea plant. Okay, now what is netonogamy? So it is a type of self-pollination in which the pollen grains are transferred from the anthers to one flower, anthers of the one flower to the stigma of another flower. So I've already told you the pollens can be transferred to the same flower, okay, or they can be transferred to the stigma of another flower, which are born either on the same plant or a genetically identical plant. Okay, this was clear. So first case was the transfer of pollens to the stigma of the same flower. Second is to the another flower, and then within the another flower, it can be, the flowers can be present on the same plant or different plant which is genetically identical, or we can say transfer to the stigma of the flower that are genetically identical. So you know, this reproduction takes place within genetically identical organisms. That's why we are writing genetically identical plant. Okay. Now let's talk about the significance of the self-pollination. First is it maintains purity of the race. Okay, because here there is no mixing of the two genome. That's why the purity is maintained. Okay, because there is no mixing of the, you can say, genome or the genes. Okay, here what is happening? The pollination is happening within the genetically identical individuals. Okay, or the flower that belongs to the, or the flower that has same genetic material or the similar genetic material. Clear? So there is no mixing. That's why the plants that are produced, that will be pure. All right. Now next is, it also maintains the superiority of the variety once developed. Okay, so this is also significance. So because of self-pollination, the superiority of the variety is maintained. Okay, so what happens here? There is no mixing. There is no mixing of the other genome. So if a superior variety is created, then superiority will be maintained. Means the superior characters. So superior characters can be maintained. Maintained means there is no loss of the character in the next generation. So how this is maintained? Because there is no loss of the characters in the next generation. Okay, why there is no loss? Because there is no mixing of the different genomes. Okay, that's why the superiority is maintained. So suppose by the process of plant tissue culture, you make a very superior variety, and then this superior variety can be maintained. Okay, this is known as maintenance of the variety. It can be maintained by self-pollinating these varieties. All right, we have to self-pollinate it. That's why the characters will be maintained and the superior variety will be maintained from one generation to the next generation. Now another one is the cross-pollination. So pollination are two types, self-pollination and cross-pollination. Now we will talk about the cross-pollination. Now it is the process of transfer of the pollen grains from the anther of one flower to the stigma of another flower born on a different plant of the same species. Clear? So this is known as cross-pollination. Clear? Here what is happening? Again the pollen grains have been transferred from the anther of one flower to the another flower. Okay, to the stigma of another flower, and that flower is born on a different plant, okay, which belongs to the same species. Clear? In the case of self-pollination, what was happening? The pollens were being transferred from one plant to another plant, okay, and that flower was born on the same plant or on another plant that was genetically identical to that plant. Okay, so this is the difference. Here a different plant is involved which belongs to the same species. Now this is the flow chart showing you the types of pollination. The first one is the self-pollination, another one is the cross-pollination. Here what is happening? The flower pollinated by the pollen grains of the same flower or of the another flower born on the same plant. Okay, it is of two types, autogamy and heterogamy. Autogamy, where the flower is pollinated by pollen of the same flower, and heterogamy, flower is pollinated by pollen grains of another flower born either on the same plant, okay, or on a genetically identical plant. Whereas what is cross-pollination? Okay, flower pollinated by pollen grains or another flower born on the different plant of the same species. Now it can be of two types, allogamy and xenogamy. Okay, so you don't need to go in detail, you have to just remember it is of two types, allogamy and xenogamy. Okay, so this is a flow chart showing you the types of pollination. Clear? Now the transfer of pollen grains. How the transfer of pollen grains takes place? So it takes place by various ways such as biotic as well as abiotic. So there are two modes of transfer of this pollen grains from the anthers to the stigma. Okay, one are the biotic ways, another one are the abiotic methods. Clear? Now let's talk about these abiotic factors and biotic factors. So abiotic factors. So this column shows you the abiotic as well as the biotic factors, and here are the technical terms that are related to this abiotic or the biotic factors. So wind, or you can say pollination by wind, this is known as anemophily, and pollination by water, this is known as hydrophily. So here the pollen grains are transferred through wind, and in hydrophily the pollen grains are transferred or they are carried by water. Okay, so you can write here they are transferred. Okay, transfer takes place or they are carried by wind or water. So these are both the abiotic factors, and let's talk about the biotic factors. Okay, so first one is the insects. So pollination by insects is known as entomophily. So entomo word is related to insects. Then pollination by birds, this is known as ornithophily. Then pollination by bats also takes place. So this takes place, okay, so and this is known as chiropterophily. So pollination of bats takes place in chiropterophily. So plants which are of very large size, they are pollinated by bats. Okay, example is Rafflesia plant. Now pollination by snails also takes place. So pollination by snails, this is known as malacophily. So these are all the biotic agents that are involved in the transfer of pollens from one flower to the stigma of same plant or same flower or the another flower. Now what is the significance of cross-pollination? So it increases in the yield as well as the adaptability. Okay, so increase yield is yield means the output. The output is increased because due to cross-pollination, what is happening? There is a mixing of the genes, all right, and a hybrid is created, and this leads to the increase in the yield, and it also helps in the adaptability. Okay, means adaptation to the environment. Now if the plant is more adapted to the environment, the yield obviously will be more. Okay, now second is it eliminates the defective traits and produces the new varieties. Okay, so all the defective traits these are lost, and what are selected? New traits which are beneficial for the organisms or the plant, they are selected, and a new variety is produced. Okay, it also leads to the hybrid production. So I've told you hybrid are produced here. Okay, so because what is happening, the two parents that are involved in the cross pollination, they are not that much genetically related. So both of the parents, they have some special characters, and those special character helps the plant to adapt to the environment. So these characters are then selected, and in the hybrid and a hybrid is produced, and this hybrid will have better yield and it will be more adaptable to the environment. Okay, and the defective trait will be lost. Okay, and by the hybrid, if the hybrid is formed, then you can say a new variety is also formed. Clear? This hybrid will have better yield, okay, because it will take all the good characters of both the parents. Clear? So usually it takes the good characters only. Now let's talk about the fertilization in a flowering plant. How a fertilization takes place in the flowering plant. Okay. So before fertilization, what was the process? Formation of gametes. Then that is known as gametogenesis. Then after the gametes are formed, next process is the fusion of the gametes, which is known as the fertilization. So fertilization is the process of fusion of the male gamete with the female gamete, and the process of formation of the male gametophyte. What is the male gametophyte in case of plant? So pollen grains, these are known as the male gametophytes. Okay, so the formation of the male gametophyte means the pollen grain. In case of plant, this is known as microsporogenesis. This I've told you briefly in the previous lecture. Initially, we have the microspore mother cells which are diploid. They undergo meiosis. Okay, and what is produced? Microspores. Microspores are produced, okay, which are haploid. Then what happens from these microspores? Pollen grains are produced. Pollen grains will be produced, which will be again haploid. Okay, so this is the process of microsporogenesis. Clear? Then what happens here? The process of formation of the female gametophyte. So what is a female gametophyte? Embryo sac is known as the female gametophyte. Okay, so female gametophyte in case of plant, it is known as the embryo sac, and the formation of the female gametophyte is known as megasporogenesis. Clear? So what happens in megasporogenesis? There is a megaspore mother cell, okay, which undergoes meiosis. Meiosis is known as reductional division. So this is a diploid. Okay, then after meiosis, megaspores are produced. Okay, a tetrad of megaspore is produced, means 4 megaspores are produced. So this will be obviously haploid. And then after mitosis, what will happen? It will give rise to the embryo sac. Okay, we'll discuss about the embryo sac in the upcoming slide. So this is known as megasporogenesis. This is clear? What are microsporogenesis? What are megasporogenesis? Now let's talk about this. Now the process of pollination, it occurs due to which the anther gets stuck up to the stigma. Okay, here the anther gets stuck up with the stigma. So this is how pollination takes place. Okay, so if the anther gets stuck up with the stigma, then what will happen? It will release the pollen grains. So this should happen during the self-pollination, anther getting stuck up with the stigma. Now after reaching to the stigma, what will happen? The pollen grains, they develop a pollen tube. Clear? And through this pollen tube, the male gametes will be transferred, and one of the male gamete will fertilize the egg cell. Clear? So if this is the pollen grain, then what will happen? It will germinate when it will land on the stigma, and then this pollen grain, it will form 2 male gametes. Okay, so these are the 2 male gametes which are formed, okay, within the pollens or the pollen grains. Okay, so each pollen, they give rise to 2 male gametes. Okay, 2 male gametes are formed. Alright, so this is what it's saying. After reaching the stigma, the pollen grains, okay, they germinate and they develop a pollen tube, and this pollen tube growth through the length of the style from where it reaches into the ovule. Clear? So what is happening here? So let's draw the structure of a pistil or a carpel. So this is what? This is the stigma. This is the style, and this is the ovary. Okay, and within the ovary, ovules are present, and within the ovule, the embryo sac will be present that consists of the egg. So here, suppose a pollen grain lands here, then it will germinate to form the pollen tube. This pollen tube will travel through this style. Okay, and then this will reach here, and then it will finally enter into the ovule. Okay, so ovule you can draw like this. So here, suppose this is an ovule. So the pollen tube will germinate, and it will travel through the style, and then it will enter the ovule, and within the ovule embryo sac is present, which consists of the egg cell. Okay, and the two male gametes are released within the embryo sac, and one of them fertilized with the egg cell. Clear? Now let's move on. Now the pollen tube, it comprises of two male gametes, which is later on released in the embryo sac through an opening called as the micropyle. Clear? So let's draw a rough sketch of an embryo sac. So this above region is known as the chalazal. So this is the chalazal end. This is the micropylar end. This is the micropylar end. At the chalazal end, you have the three antipodal. Okay, so these are known as the antipodal cell. Okay, which are haploid. In the center, you have the polar nuclei. Okay, these are known as the polar nuclei. And at the micropylar end, you have the egg cell as well as the synergids. So the above one, the center one, is known as the egg cell, and these two, these are known as the synergids. The pollen tube will enter from the micropylar end. So here you can see pollen tube. This is the micropylar end. So pollen tube will enter here. Okay, so this embryo sac is within the ovule. So this pollen grain will enter through this micropylar end. This pollen tube, it will consist of two male gametes. So these are the two male gametes. Okay, so these two male gametes enter into the embryo sac through the micropylar end. So it is what is saying. Pollen tube, which comprises of the two male gamete, it is released into the embryo sac, okay, through an opening known as the micropyle. Okay, so this, you can say, it is the pollen tube. Okay, that is formed by the pollen grain, and this is the structure of the embryo sac. Okay, this is the structure of the embryo sac. Clear? Now one of the male gamete, it fuses with the egg, okay, to form a diploid zygote. So one of the male gamete, it will fuse with this egg cell to form the diploid zygote, and the other male gamete, it fuses with the polar bodies to form the diploid nucleus. So you can see these polar bodies, these are also haploid. Egg cell is also haploid. Synergids are also haploid. So these polar nuclei, first they will fuse together. Okay, they will fuse together, then they will form a diploid structure, and this male gametes, one of the male gamete will then fuse with this diploid structure, and then a triploid nucleus is formed, which later on develops into the endosperm. Okay, so one of the male gamete, it fuses with the egg cell, and this will form the zygote, and the other male gamete, it will fuse with the, you can say, the two polar bodies. Okay, it will fuse with the polar bodies or the polar nuclei. Okay, then what it will form? It will form a triploid nucleus, which later on develops into the endosperm, which provides nutrition to the developing embryo. Okay, so one of the male gamete. So you can write here, there are two male gametes. One of the male gamete, it fuses with the egg cell, and the other male gamete, it will fuse with the polar bodies. Clear? Now this is known as double fertilization. Here the fusion of gametes is taking place twice. So the process of fusion of one male gamete with the egg cell and the other male gamete with the polar bodies, this is known as double fertilization. So this double fertilization is seen in the plants, or it is seen in the higher plants such as angiosperms. Angiosperms are flower bearing plants. Clear? And the fusion of the one male gamete with the two polar bodies. So there were two polar bodies to form the endosperm. This is known as triple fusion. Why? Because it involves one male gamete and two polar bodies. Clear? Now what is an endosperm? Endosperm is meant to provide nourishment to the developing embryo. Okay, now you know the whole process of double fertilization. What is happening? One of the male gamete is getting fused with the egg cell, and the other male gamete, it fuses with the two polar bodies. Clear? Now after fertilization, what happens? The sepals and petals, they will fall, and the zygote, they undergo a series of mitotic divisions, okay, to form a multinuclear embryo. Alright, so first process was formation of gametes, then second was the fusion of the gametes, okay, which is known as fertilization. Then after fusion, the zygote is developed, and then zygote undergo mitotic divisions to form multinuclear embryo. Now at maturity, wall of the ovules, okay, they change to seed coat. Okay, so ovules, you should remember that ovules, after fertilization, they develop into the seeds, and the ovary, they develop into fruits. Okay, now at maturity, wall of the ovules, they change into the seed coat, okay, of which the outer one is hard and is known as the testa. So seed coat has two layers. The outer is known as the testa, and the inner one, it is known as the tegmen. Now after fertilization, ovule develop into the seed, and the ovary wall changes into the fruit wall, which is known as the pericarp. Okay, fruit wall is known as the pericarp, and the ovary, it changes into the fruit. Alright, this is clear. So this is the image showing you the fertilization or double fertilization in the flower. Okay, here you can see this is the embryo sac here. Alright, first we can see here, these are the anthers. Okay, this is the filament. These anthers, they will produce what? They will produce the pollens. Okay, so pollens contains what? They contain the male gametes. Alright, and these male gametes, okay, they are formed within the pollen grains, and then you can see this is the pollen tube. Alright, pollens, they germinate when they land on the stigma. Okay, so stigma, then the pollen group, they travel through the style. So this is the style, and here you can see the pollen group tube, it enters into the embryo sac through this micropylar region. Okay, so this is the micropylar region. Okay, through the micropylar region, the pollen tube will enter into the embryo sac. This is the ovule, which contains the female gametophyte. In the female gametophyte, this is known as the embryo sac. Okay, female gametophyte is known as the embryo sac. Okay, within the embryo sac, you can see, I've already shown you what is present. The egg cell is present along with the synergid at the micropylar end, and the antipotals, they are present at the chalazal end. Okay, so here when the pollen tube, it enters through the micropylar end, where the egg cell as well as the synergids are present, then one of the male gamete will fertilize the egg cell, and other male gamete will fuse with the two polar bodies, leading to the formation of the endosperm. So this is an image showing you the double fertilization in the flower. Again, I will repeat, anthers will produce the pollens. These pollen grain will produce two male gametes. Then when the pollen land on the stigma, so this is the stigma, this upper part is the stigma, then this is the style, and this is the swollen part known as the ovary. Ovary consists of ovules, and okay, and within the ovule, what is present? The embryo sac. So this one is the female gametophyte known as the embryo sac. Clear? Now, one of the male gamete will fuse with the egg cell, another will fuse with the two polar bodies. Now let's understand the difference between self-pollination and cross-pollination. So pollen grains are transferred from the anther to the stigma of the same flower. During self-pollination, this is known as the autogamy, or if it gets transferred to another flower of the same plant, this is known as the heterogamy. Clear? Now what happens in cross-pollination? The pollen grains are transferred from anther of one flower to the stigma of another flower born on a different plant of the same species. So this is known as the allogamy. Clear? So now you know about all these terms. Autogamy, neotogamy. Autogamy is the transfer of the pollens from the anther to the stigma of the same flower. Neotonogamy is the transfer of the pollens to the another flower which is present on the same plant. Okay. Allogamy, transfer of the anthers of one flower to the stigma of another flower born on a different plant but which belong to the same species. Now both the anther and the stigma mature at the same time. Okay, so this is a requirement for self-pollination. Both the anther and the stigma, they should mature at the same time. Okay, otherwise if they don't mature at the same time, then it has to undergo cross-pollination, because self-pollination will be inhibited if they both, anther as well as the stigma, they don't mature at the same time. Then only stigma will be receptive for the anthers if they mature at the same time. Now cross-pollination, the anther and the stigma of the flower generally mature at the different time. Now let's talk about self-pollination. It can occur even when the flowers are closed. Okay, even so, the floral parts, they are not exposed to the environment. So even if the flowers are closed, okay, so in some plants the flowers are closed only. So here self-pollination can take place, and the anther and stigma should mature at the same time. Whereas in the plants where the flowers are open, means they are exposed to the external environment, here cross-pollination will take place. Okay, so in open flower, cross-pollination; in closed flowers, self-pollination usually takes place. Now, external agent is not required for self-pollination. Clear? This I've already told you. The anther can stick up to the stigma. Okay, this helps in the this happens in the self-pollination, and there is no external agent required. So there is no requirement of any carrier for the transfer of the pollen grains. But in cross-pollination, an external agent is required, which can be biotic or abiotic. This is essential for the cross-pollination. Now, it is economical for the plant, means less energy is being spent for the self-pollination, where cross-pollination is not economical for the plant. Okay, because plant has to produce a lot of pollen grains as well as the nectar, okay, scent and bright color, petals also. So corolla are the collection of the petals. So plant has to spend a lot of energy, okay, and food resources for the production of large number of pollen grains, because here the pollen has to be transferred to the flower of another plant. Okay, so they can be lost and they have to travel a lot of distance also. That's why a lot of pollen grains are produced. Nectar are also produced. Okay, nectar is produced. Why? Because there are some biotic agents such as bees. So they feed on this nectar, and when they feed on this nectar, or when they suck this nectar, then they transfer the pollen grains also. They produce scents also. Scent is to attract again the insects, as well as the bright color of the petals is also required for attracting the insects as well as the animals. Now the self-pollination ultimately results in progenies which are pure lines, or they are homozygous. Okay, because here the transfer of the, you can say, the pollen grains are to the same flower, okay, or to the another flower which is genetically similar to that plant only. Okay, so that's why the plants which are produced, or the progeny which are produced, they are pure lines, means they are homozygous for a particular character. Clear? Now cross-pollination, it produces offspring which are hybrids. Okay, that is heterozygous, and they show variation in the characteristics. So the progeny which will be produced, it will show variation, and the progeny, you can say, we can call it as hybrid. Okay, whereas pure lines are formed because of the self-pollination. There is no mixing of the genome here, or the genes here, whereas here there is mixing. 7th point, it cannot eliminate useless or harmful characters. Okay, but whereas cross-pollination, it can eliminate useful or the harmful characters. So this hybrid which is formed, this will be, you can say, it will contain all the good characters, and the harmful characters will be eliminated, and the hybrid will be better adapted to the environment, and it will have more yield. Okay, now self-pollination, highly useful characters get preserved in the race. Okay, whereas in cross-pollination, useful characters cannot be preserved in the progeny. Okay, because these useful characters may be lost. The next point, self-pollination does not introduce any variations and hence the offerings are unable to adapt to the change environment. Okay, so this is a negative point for self-pollination. So there is no introduction of the variation because the pollination is happening between genetically similar plant, okay, and the offerings. So there is no mixing of the genome in the offerings, and whatever happen, if the environment changes, then that plant will be unable to change in the environment as there is no mixing of the genome. Clear? Whereas in cross-pollination, it introduces the variations in the offerings, and these variations make these plants to adapt better to the change environment for the struggle for the existence. Okay, so these variations are brought about in the offerings due to the cross-pollination. Clear? And because of this variation, the plant is able to survive in the changing environment. Clear? So there is a struggle for existence. Okay, clear? And the better adapted individual will survive. Clear? Now, immunity of the race towards the disease falls in the succeeding progeny. Clear? Whereas immunity of the race towards the disease is usually marinated in the succeeding progenies. Okay, means it increases in the succeeding progenies because the characters are being selected as the environment is changing. Okay, and these characters help to produce the variations as well, and these variations help to adapt these organisms to the changing environment. The next point is yield of the plant gradually falls with the time. Okay, because the plants, okay, they cannot adapt to the changing environment, so obviously the yield will fall, whereas the yield of the plant usually does not fall. Okay, so hybrid which is formed, that will have better yield than the parents. Clear? Now next point, self-pollination never helps in the production of new varieties and species, whereas cross-pollination is the mechanism of producing new varieties as well as species among the plants. So these were some of the difference between self-pollination and the cross-pollination. Okay, so in this lecture we have studied about what is cross-pollination, what is self-pollination. Okay, then we have talked about what are the methods of the self-pollination as well as the cross-pollination. All right, so in the next lecture we'll be solving some of the questions that are related to these topics. Okay, hope you enjoyed this session. Thank you very much.